Hello, my name is Jessica Hoyt, and this presentation will delve into the research efforts of several scientists, and particularly, I will focus on the findings of Paul Gabrielson and his colleagues. Paul Gabrielson is an adjunct assistant professor in the biology department of the University of North Carolina at Chapel Hill. He collects algae specimens and preserves them for scientific studies. The collection of preserved plants is known as an herbarium. Recently, he published an article about red algae. In this publication, Gabrielson and his colleagues worked to sequence the DNA of past specimens and determine their correlation to today's collections. Thanks to well-kept herbarium specimens, we are able to study past collections. What is DNA sequencing? Firstly, we should determine what DNA does. DNA is the coding that makes every living organism different from one another, and it tells each cell what to do. DNA is built of little blocks in a particular order. These blocks are attached to two different strands wrapped around one another, which is where the helix structure comes into play. When scientists sequence DNA, they're using different techniques in a laboratory setting to determine the order of the DNA blocks. By mapping out the DNA sequence, we can better understand how the subject has evolved throughout time. To study red algae, they cracked open the old herbarium samples from as early as 1902. They also collected recent field samples to compare their sequenced DNA to. The old samples are labeled as the same species of the current ones. However, they found that there are two samples which they cannot identify and thus should be redefined in the herbarium. Now, scientists may be able to study the evolution of marine species with a closer eye and connect past species to the present species. Studying the past is important in all subjects, not just history. Thank you so much for your time. That's all I have for you today.